right. So muscle, just like any other tissue, has to do with how you cut it, right? And this is a good slide, and it's named, shockingly enough, is named smooth muscle. So the thing that we want to look at here is, it depends on how you cut it, right? Just like anything else, how you cut it. And this one is good because you have some that's cut, what's called a longitudinal cut, and some that's cross cut, right? So you have longitudinal cut, cross cut, smooth muscle. So, what are you looking at, right? So, smooth muscle, it's not really heavily organized. We're going to zoom in on it a little bit so you can see some stuff in there. The thing to remember in smooth muscle is that you have this kind of a fusiform shape that you're looking for, right? Which we've used that term tons of times in here. Some kind of yuck on the slide there. And you can see these fibers, if you look close, you zoom in and out, you can see these little fibers that go about that far, and that's about all there is to them, right? These little fibers in there, and where are the nuclei? They're in the middle of them, right? So the nuclei are just everywhere. They're not pushed anywhere. They're in the middle of the tissue, and they're in the middle of the fibers, shall we say. And the fibers are all these little things about this long, right? They're not very big. So that's kind of the it's a longitudinal cut. And then the cross-cut one, if we slide over, look at that one, they are in little bundles per se, but they're not really highly organized. I mean, they look pretty nice, and again, the nuclei are in the middle, as you can see, and you can see the different layers of the different peritonia, right? Not peritonia, mycia, right? What are the 3 mycia? They were on the test, right? It went live, everybody can see it, right, I hope? Nobody's having any issues with the test, right? It's on Blackboard, you need to print it. Remember I said print it, actually write on it and turn it in on Monday, right? I did set it up to go live right after class, or right after I put it up there, so it should be live. Anyway, so what are the different mycia again? Perimycium, epi, endo, right? All the standard prefixes, right? So epi on the outside, peri in the middle, endo on the inside, right? And you can see them, right? You can see there's a layer there, there's another layer, here's a little layer there, right? So you've got these different layers of different connective tissue capsules or sheaths around the outside of it, right? All right. Fairly easy, smooth muscle, and okay, I'm gonna go back to my lowest power here again. Where's the only place you're gonna see this? Where do you see 2 layers of smooth muscle, one longitudinal, one cross-cut?
Stomach.
And 2, say that? Intestine, very good. In the intestine, right? You're gonna see that throughout the intestine. And that's what this is a little piece of an intestine is what it is. I'm trying to see if I can tell which region it is. No, I can't. There's not enough tissue there to tell. Anyway, we'll come back to that. All right, smooth muscle, very easy. Next one is a skeletal muscle.
Yes.
Now there are a lot of different slides of skeletal muscle. Some are better than others. So this one is skeletal muscle, CS and LS. It's an older slide. It's okay. Specifically, anybody who can tell me what tissue that is? What organ that is? So the one first thing here, this is the muscle down here, all this stuff. And notice it's going all different directions, right? But then you have this epithelial layer up here, right? And I'm sure everybody knows what type of epithelium that is.
Loosely arranged dense connective tissue. No, it
It's
doesn't.
stratified squamous Anybody wanna make a guess whether it's keratinized or not?
Non-keratin.
Non-keratinized. Stratified squamous, non-keratinized. That only happens in a few places. Say that out loud. Tongue, very good. He pointed to his tongue for those of you who didn't see it. He actually stuck his tongue out and pointed to it. So this is the tongue. One of the most versatile muscular organs in your body, right? You have more ability to manipulate your tongue than you do your fingers, right? So this is the muscle here. This is glandular here, by the way. That's glandular. This is muscle over here, right? So skeletal muscle, lots of it. So let's look at some of this and see if we can get a good image of what it looks like under high magnification, because we want to look for the striations, right? You want to see the striations in the muscle. Are they easy to see in here? Do you see striations?
Yes.
Kind of. Why don't they look like the classic pictures of striations like in your lab image?
Is it the same?
This is not a true longitudinal cut. This is actually what's called a sagittal cut. So the tissue is going like this and it's cut like that, right? It's not a true cross cut, it's not a true longitudinal cut, it's kind of a diagonal cut. And so you're seeing these weird little wispy lines that aren't really showing you what you want to see, right? So let's see if we can find somewhere where we have a true true longitudinal cut. And you may or may not see it in this. Oh, there's a little piece of it there. There's a little bit there that's fairly true longitudinal cut. See if I can show you what I'm talking about. It doesn't show up really well. But as I said, so this is okay. It's not perfect by any means. We can find better skeletal muscle, much better skeletal muscle. So what I want to do to show you a better skeletal muscle side is I want to go to one of the slides that we love, right? Which is one of the slides that we love that has all these different things on it that we want to look at. Which one do you figure this is that I want to go to? Nobody? You're not having any. See if I can find it. It was a slide I used for the test. It's the fibrocartilage side. The one with the bone in it, right? What are you doing? What are you doing?
My coworker sent me a message, so I was trying
So you're listening to messages while I'm talking? I know that I'm not very exciting lecturer up here. All right, so this one we're gonna look at the skeletal muscle here. So that's this stuff, right? This is the skeletal muscle here. Notice how pretty that is, right? Much brighter color, different color, right? It's red instead of purple. That's because of the stain they're using. And how does this look comparatively? Is this better or worse? No? Let's go up one more and see what it looks like. How about now? Those definitely look like the striations you're used to, right? And you can actually see those striations in there. Now on this one, like right here, this is a really nice little piece of tissue here. Up there on top, that top section up there. Now the only bad thing about this stain is that it doesn't show off the nuclei very well. But the nuclei are along the edges of the tissue. They're not in the middle of it. In skeletal muscle, the nuclei are smashed over to the edge. The mitochondria are smashed over to the edge. All the organelles are smashed to the edges. Nuclei are going to be bigger, mitochondria are going to be smaller. That's the only real difference in what you're going to see, but they're relatively small.
Our picture doesn't really have them towards the edge.
You did what now?
Our picture, the mitochondria and the nuclei, it doesn't really have both of them near the edge. It has one of them sort of near the edge, but
Well, let me go back 1 second. No, I was going to just say this actually. If you go to a section where it's cross-cut, like this one, you can actually see it better, right? It's easier to see where the nuclei and mitochondria are on a cross-cut section because it's much more distinct, right? So if you zoom in on a cross-cut tissue, that's not true cross-cut. That's not a true cross-cut, by the way. It's close, but not quite. This isn't bad, right here. This is okay. All right. See if I can zoom in. No, that's as high as I can go. Let's go down one. Let's see if that actually helps. Not bad. All right. So in crosscut, you can actually see those bundles a lot better. Let me see if I can find a different one that might be better. Ooh, this one might be good. This is a longitudinal section, but this one will be pretty at least, I think. Sorry, excuse me. Oh. How's that for looking? That might show you the mitochondria and the nuclei, right? So pretty nice. That's a really nice, clean image of skeletal muscle. The only thing I can say bad about it is it looks wavy, which is kind of weird, right? Other than that, you can see the nuclei, you can see the mitochondria, you can see all those things, right? No? Maybe?
Where's the mitochondria?
Where's the mitochondria?
Yeah.
All right, so nuclei are big, right?
Yeah.
So nuclei are these big things, right? Mitochondria are gonna be small, like so, and little tiny things, not very big. They're not very big like this one and that one. They're little and small, but they're pushed off to the side as well.
What causes the wavy pattern?
All right, so your muscles are attached to something end to end, right? Like we were talking about how it causes the movement, right? Well, what happens to a rubber band when you take off how it's stretched?
Scrunches together.
It scrunches together, and that's where you get that waviness, is because it's not connected to anything when they made the tissue, embedded the tissue.
Gotcha.
All right, well, that's a really nice slide. That one is called voluntary muscle LS, and it says in parentheses striations. It's a really nice slide. So if you get a chance, look at it. It's a nice slide for skeletal muscle.
Thank you. Thank you.
All right, what's next?
Cardiac muscle.
Cardiac muscle.
Heart. Okay, let's look at some heart muscle. Let's see, how about this one? It's okay. Don't love it. Let's see what else we got. How about this one? That's better. Okay. So this is muscle cardiac section is what it's called. So it looks kind of like skeletal muscle, and it looks kind of like smooth muscle. It's kind of a weird cross between the two, right? It is striated if you look at it closely. Can you see the striations? Do I need to go higher? Can you see the striations down? Look right here, right here, right? You can see, very faint, but they're there. Here, you can see them, right? If you focus in and out, you would see them better. So it's striated. It's also branches. It's one of the few muscles, in fact, it's the only muscle we have that does branching. The other thing that you have in cardiac muscle that you want to see is these connections between the ends of the muscle cells, right? And what do we say they're called? It's one of those pronunciation things, right? How do you want to say it? I don't care which one it is, as long as you know how to spell it, right? It's either intercalated or intercalated, either way you like. And that's the discs, and you can see them here. Anybody see them? So, for example, here's one here. Here's one here. Right? Seeing them now? Where else is one? There's one over there. There's one here. One here. One there. One there. Any others? Probably a ton of others. Just one up there. A lot of them, right? Now there are stains that are specifically for intercalated discs to make them easier to see. The nuclei and cardiac muscle are in the middle of the muscle tissue, so it makes it look a little bit like smooth muscle in that respect. This is going to be a very different stain, by the way. This is a metallic stain, and look at the color you get, right? Metallic stains have a very different appearance, but they do show you some other structures that you don't normally see. Very nice picture, right? Striations are very easy to see here. Nuclei are very easy to see here. And you can also see the intercalated disc very easily as well, right? Anything on that? Questions, statements, comments, concerns? Nope? Okay. So, the last thing we're going to look at in the heart has to do with has to do with heartbeat, right? The heart is one big unit. It is functionally one large cell, or one large structure, if you will. In the heart itself, there's nothing but heart. That's it. So how does the impulse, so you send the contraction impulse to the heart. How does that impulse go from one part of the heart to the rest of the heart? And there is a modified muscle cell that does that work. And what's it called? Purkinje fiber, right? So let's look at Purkinje fibers. And Purkinje fibers are easy to see. So this is hard tissue. That's my lowest power. So we're just going to wander around until we find something that doesn't look right. So it's heart muscle, excuse me, heart muscle. And we're going to go around until we find something that looks weird, right? I hate that what's in focus for me is not in focus for you. All right. Anybody know what this is, by the way? It's exactly right what it is, adipose. Why is there adipose right on the outside of your heart?
You need a lot of energy storage, that way you can have the easy access to it.
It is a high energy consumption structure, right? And so almost everybody has some adipose around their heart on the outside of it. What's the problem? Too much, right? Just like anything else, too much of anything is a bad deal, right? So you get too much lipids in there, too many lipids in there, it becomes troublesome in many ways. All right, so let's get away from that and go look at Purkinje fibers. Did you find anywhere where there are weird spots? How about if we look, let's see, where can I pick? I don't care where I pick, let's pick somewhere. Anywhere looks good to me. All right, let's do this view. There's a big couple of weird things on their slide, but don't worry about those. There we are. Now we're in focus. Don't worry about this. Don't worry about that. That's just weird stuff, right? Don't worry about it. So, where do you see some cells that look weird?
The dark red ones.
So, where they look weird is specifically on the periphery, right? On the edges. Here and here. Does everybody see why it looks different? I'm going to zoom in on to give you a better view, but notice how it looks lighter in shade, and it looks like there's almost gaps in it there, right? That's the key distinction, and it usually is on the edges, okay? So let's zoom in on it a little bit. I'm going to zoom in specifically on this area here because it looks really nice. So now we look at it here, and I want to put it like that so you can see. All right, that's not bad. Okay, let's look at that. So this is normal cardiac tissue. This is cross-cut, by the way. Normal cardiac tissue up here. This is Purkinje fibers down here. And notice how you have like this clear little spot in the middle of these fibers, right? That's the distinctive feature of a Purkinje fiber, is that weird little central clear spot. So what is a Purkinje fiber and what does it do? It's a modified muscle cell, modified cardiac cell that conducts a nerve impulse. So it has changed its job from contraction to nerve or electrical conduction. So they're the things that carry the impulse from the top part, the atrium, down into the ventricles. But they are still muscle cells, they just have been modified to change their job. And it's not actually a big leap, to tell you the truth, right? Think about how a muscle contracts. The nerve, the charge runs down into the muscle cell, right? And goes in there and does changes to what's going on, right? So it's already conducting a signal along the surface. All it has to do is not go down into the cell and cause the contraction to occur, and just continue along the surface, right? It's not a huge change. But you can see lots of them there, and they're fairly easy to spot. All right, any questions on Purkinje fibers? I know you can probably guess the name of the slide. Purkinje fibers, all right. And it actually says human heart on it, so, all right. All right, next we're going to go away from muscle, we're going to look at nerves, right? So first we're going to look at nerve fibers and how they work. That's what the name of the slide is, nerve fibers. This is another metal stain. So let's zoom in and look at a nerve fiber and look at how it looks. I really don't care where we look, it's all fine. How's that look? Pretty wild looking, right? So there's several different things we need to look at here. So here's one of those nerve fibers. Doesn't look like a continuous thing, does it? Looks like it's disjointed, right? The reason for what this is that you have several parts. This is kind of like, think of like an electrical wire. You've got the inner wire that's actually the wire, and then you've got this plastic coat around the outside, right? It's very similar here. You've got the nerve fiber, which goes in the middle, and then you've got what's called a myelin sheath that goes on the outside of it, right? Now the problem is that the electrical charge has to go along the outside, but myelin is primarily composed of lipids, and what do lipids do to electrical charges? They do not conduct electrical charges, they insulate it, right? So what does that make the nerve charge do? So if it can't go along the surface because of the myelin, it has to jump from one spot where there's no myelin to another spot where there's no myelin. And what's the scientific word for jumping?
Saltatory.
Saltatory, right? Salt. Or stalt, take your pick. Saltatory movement. So it jumps from one spot where there's no myelin to another. So where's a spot there's no myelin? And that is one of these things. See how it kind of bows out and then goes in, bows out and then goes in on both sides, and then there's this little weird little spot in there. That is the spot where there's no myelin. It's not very big. It doesn't have to be very big. But it's going to jump from one of these to the next one, right? These little structures here are called nodes of Ranvier, right? Nodes of Ranvier. Now there are other structures in here as well, right? And if you look here, you can see these little dark lines that go across, right? That look funny, right? So when you're laying down the myelin, it only can lay down the myelin as big or as wide across as the cell is. Or a little bit wider than that, but still there's a maximum distance it can lay down the myelin. So these little dark lines are the length of the cell that's laying down the myelin. So that's one myelin group versus another myelin group. And so these lines that are between these myelin layers are called incisures of Schmidt-Lannerman, right? That's just the edges of each little myelin packet. Now, if you get the right piece of it, you can actually see the internal nerve fiber. And actually, you can kind of see it here. Yeah, you can see it right there. Okay. You can see the internal nerve fiber right there, right? And that's the myelin on the outside, right? You can see the nerve fiber going right through the middle there, right?
Could you point against the incisors?
Incisures?
Yes.
Well, here's one here, there's one there, there's one there, there's one there. Little dark or light lines depending. Here it looks like a light line, there it looks like a dark line, it depends. But it's those little weird little lines that go across. The nodes are always going to be this big bulb looking thing, right? It always has that big bulb look to it. Let me see if I'll show you another node. Nodes are fairly easy to see and they're kind of the fun part of it, right? Let's see, how about that one? Yep, you can barely see it, but this there, right here.
And those nodes have no myelin connecting.
The spot that doesn't have any myelin is not very big, it's kind of in between them, right?
Gotcha.
How about that one? So the spot that doesn't have any myelin is this spot right between them, that's it. It's not a very big amount of space that doesn't have myelin, but it's a small space that works. Okay. Nerve fibers, pretty straightforward. That's here's a different slide of the same thing, basically. There are different versions of each one of them. There's no real difference here. That's very similar, right? Not a lot of difference there. You can see the nodes, you can see the myelin and scissors, you can see all that, right? All the same stuff, right? There's a nice node up there. All right. Let's look at a nerve. Slide says nerve mammal. That's what the slide says. So this is a, excuse me, it's a crosscut nerve bundle. And this is where you can see the mycia real well. Actually, It's not muscle, it's nerve, so it's neuria. Same difference, right? But you got the same three layers here, right? You've got epi, peri, and endoneurium, right? Instead of mycelium. Epineurium, perineurium, and endoneurium. Same three layers. So, epi, peri, endo, right? Now look at this and just think about what we're looking at here for 1 second. So this is a nerve bundle that's in your body, right? They're not very big, obviously, but each one of these, right? Here, just look at this little bundle all by itself. This is just one little piece of a nerve bundle, right? Each one of those little circles there is a nerve fiber. So how many nerve fibers are in that bundle alone? Hundreds of them, right? And there's how many of those in here? And that's a little one compared to this bigger one, right? And you have how many of those running through your body? These are like the cables that connect one part of your body to another, right? This is communication. You have two different communication systems in your body. I've mentioned that before, I believe, right? One of them is a fast communication system, one of them is a slow communication system. Which one is this? It's the fast communication system. So, nerve system, fast and temporary. Endocrine system, slow and permanent, right? Mainly permanent. So again, why do we have those? Why do we have these neuria, right? Why do we have these layers of things around these things? You have a connective tissue capsule or layer around each individual nerve fiber. Why do you do that? Why do you have it? How does a nerve conduct information? Electrical charge. If you didn't have that coating around it, if this fired, what would it do to the ones around it? Cause them to fire, right? And then before you knew it, the whole thing would be firing at the same time, right? So you have to isolate the charge so that only one nerve fires. Okay. Any questions on any of that? What?
Yeah, with muscle cells, I remember you saying that they all contract at once, right?
Of one fiber.
Of one fiber, gotcha. For some reason, I thought it was the entire like peri. Gotcha.
No, actually, do I want to go back to a muscle fiber? Remember I was talking about holding the book up, right?
Mm-hmm.
Sorry, I got something stuck in my throat. So you got a muscle bundle, right? If you're using that muscle to hold something, and it's not max weight, you're not going to use the whole bundle for it, right?
Right.
Some of them would be contracted, some of them will not be contracted.
Gotcha.
And what will happen is if you're holding it for an extended period of time, those muscles get tired, right? So it'll actually switch off which muscles are contracting to keep the same distance going, right?
Gotcha.
So no, the whole bundle doesn't contract at one time, and specifically it doesn't, very seldom does it do that. All right. This is all the easy stuff. Now we're looking at the peripheral nervous system right now. Yeah, except I don't have the slide I need. Yeah. All right. So, what, I don't know what page that is. Number 5, this one. Now, the problem with this is it's a very large structure. So this is this section is titled Peripheral Nervous System, right? What are we looking at here? Does anybody know? Right. That's the spinal cord. It's the central nervous system, right? So we're not going to look at the central nervous system right now. What we're going to look at is connections to that central nervous system. Next week, we'll look at the central nervous system. Things like the brain and the spinal cord. Right now, we're going to look at the peripheral nervous system, and so things that actually connect to this structure. And that is going to be your so-called dorsal root ganglia and so on and so forth. And that's going to be over here on the edges, right? It's off to the side of it. These things, right? Dorsal root ganglia. Where does that occur? Where's your spinal cord run? Everybody knows, right? It's back here, right? So where's your dorsal root ganglia?
Right here.
It's on the dorsal side to the edges, right? So where is that? Everybody knows that you have like a furrow on your back, right? Right down the middle, there's an indentation, right? And that indentation is where your spinal cord is, right? But on the edges, there's these outgrowths, right? These thickenings, right? Where it pops out, right? Everybody know what I'm talking about? So what do you think's in there? Nothing. Fat. There is some fat in there. There's also the dorsal root ganglia there, as well as some muscles and other things in there. So there's muscle, there's dorsal root ganglia, there's a lot of things in there, but a lot of that is to protect these dorsal root ganglia. So these dorsal root ganglia are the structures that control the communication into and out of the spinal cord. And that's going to be this slide, by the way, this picture, right? Where it says B, dorsal root ganglia, right? So what you have is, you're going to kind of have to look at both slides if you can, or both pictures. The gold one is showing how, you can see the dorsal root ganglia on the sides of it, way off to the edges, right? And then you see these weird fibers or things that are running from the dorsal root ganglia into the central spinal cord, right? So notice that one goes on one end and one goes on the other end, right? One goes on the dorsal side, one goes on the ventral side. Do you know what those are? One bubble's incoming, one's outgoing. One's an efferent, one's an afferent, right? Does that make sense?
Ventral's efferent, right?
I don't know which one's which. I always get them mixed up. I've been getting them mixed up for years. Mainly because it's always upside down, which is always weird to me. You got the dorsal root and the ventral root, right? Top and bottom. And that has to do, we'll talk, next week we'll talk about what the dorsal horn is, what the ventral horn is, and which side is which. But what I'm going to look at now is I'm going to look at this dorsal root ganglion a little closer. And notice it's got a very distinctive look to it. Very distinctive look to it. And if I zoom in a little bit more, not too much, a little bit more. So can everybody see here that we've got kind of two sets of cells, right? So what you have is you have these big structures in the middle. Notice that one. Big structures in the middle. Those are cells. So you got two options, right? You got capsule cells and ganglion cells. Which one do you think? Well, that's probably the ganglion cells, right? The big ones. The capsule cells are these little guys around the outside, right? That form a capsule around the ganglion cells. For Pretty straightforward, not hard. We're gonna try to keep it easy today. How's our time going? 45 minutes. And that's with talking about dog stuff. All right. Next slide I'm gonna look at is the terminal ganglia slide there. Oof, not that one. That's horrible. That's just bad. All right, we're not looking at that one. Where's that? Where's that one slide we had of the non-striated muscle? That's probably a good one. We're gonna go all the way back to the first slide that said smooth muscle section. So if you look between the longitudinal and the cross-cut muscle sections, right in between them, every once in a while you're going to see this weird little sketch of weird type tissue here, right? Now the picture you have has a huge one going all the way across, right? A huge band going all the way across. You don't always see that, by the way. Most of the time it's much smaller and a little harder to detect. Trying to find a decent spot of one. There's not a bad one. Is there another one? Yeah, a little one there. That's probably the best one. We'll look at that one. There's a couple of them here. That's all right. So here's one here. There's one there, and there's one there, and there's another one up there. So those are these nerve endings or encapsulated nerve endings that are where they house. They're housed between the two smooth muscle layers, right? So what do you think their job is?
To make them contract.
They're controlling the contractions of these two layers of muscles, right? And we'll talk more about them as it goes. But that's just one example of where some of these terminal ganglia are. There's lots of them in your body. That's just a really easy one to spot. And they have all the standard structures. It's a little harder to see, but they have all the standard structures. They have the epineurium, they have the capsule, they have the endoneurium, perineurium, all that stuff. Is it easy to see? No. Would I ask you that on that slide? No. I would probably ask you that stuff on the one that says nerve mammal, right? The one where you can actually see them easily, right? This one, just know that it's a terminal ganglia and that is fine for now. Okay. Next, we're going to go back to the tongue. This slide says tongue vs taste buds. So we're going to look at some taste buds on the tongue. This is a nice slide. It's got also some very nice, this has all kinds of good stuff on it. This is down deep in the tongue, but look at that. That's a nice skeletal muscle there. Cross cut and longitudinal cut for skeletal muscle plus nice glands here. Everybody knows what glands are in the tongue, right? Salivary glands, yeah. Let's go up to the surface. So on the surface, let me go down one magnification here so you can actually see. On the surface, you have these things. It almost looks like teeth, doesn't it? Does it look like teeth to you, kind of? So you have these little hill-like structures, if you will. And what are those called, those hill-like structures? They're Latin for hill. I'll bet you don't know that, right? What's Latin for hill? Papilla. So, foliate, papillae. Foliate meaning like foliage on a tree or leaves on a tree, right? So this is a foliate papillae, meaning it's a hill that's got things attached to it. The taste buds are these weird round looking things, right? We've looked at them before, right? And I always think they look like little cocktail onions, right? Some of you may not know what cocktail onions are. And what do they do? What do they taste like?
Taste.
Scientifically speaking, what do they do?
Sense chemicals.
Chemoreceptors, right? Chemical receptors, right? That's what taste is chemical receptors, right? You're tasting chemicals. Everybody knows the standard statement that certain different regions of your tongue taste different things, right? And that's a generic kind of thing. In general, that area of your tongue tastes for that specific thing, right? That specific chemical per se. Anyway, and we talked about how your sense of smell is basically modified taste buds as well. Those are chemoreceptors too, right? All right, let's go on to, oh yeah, Pacinian corpuscles. All right, Pacinian corpuscles are next, right? So we're going to go, to look at Pacinian corpuscles, we're going to go to a slide that's named Meissner's corpuscles. Meissner's corpuscles in VS skin is what it is. It's one of those we can see about 50 different things in it, right? And one of those things we can see are Pacinian corpuscles. And there's one, they're huge things, right? This is my lowest power. That's one there. Looks like a cross-cut onion kind of, right? There's another one there. This one only has the two of them in it. So that's Pacinian corpuscle. Anybody know where else so you only have, oh here's another one. So this one, I'm gonna show you a different one now. I'm gonna go to a different one. So this one is called corpuscle of Vater Pacini section. The guy who discovered these structures' name was Vater Pacini, Italian. So we call them Pacinian corpuscles. So this is the other location that they occur in your body. Anybody see them? Anybody see one?
No.
They're big and strong, right? Can't miss them, right?
Those are muscles.
And what organ is that?
Would be your lung. No, probably not.
So what did you say? Say it louder.
Lung?
No. These are, so Pacinian corpuscles are pressure receptors. They're in your skin, of course, so you can feel pressure, right? This is a gland. If you look at it, think about, you got to look at what you're looking at, right? This is a gland. So what gland do you need a pressure receptor in?
The spleen.
The spleen.
No,
Spleen's
it's
not
not a
well,
gland.
okay. It's kind of a gland.
Mammary glands?
What?
Mammary glands?
Mammary gland, is that what you said?
Yeah.
No.
Like, bladder.
Urinary bladder, that'd be an interesting one. Except it's not a gland.
Oh, okay.
It would be intriguing to
I
have
was thinking of pressure. Like,
pressure receptor
I
there.
mean, doesn't it have to,
That's a different, I can talk about that if you want. Everybody knows when you're full, right, in urinary ways, right? But it's not a pressure receptor you're feeling. It's when that transitional epithelium gets stretched there's little nerve fibers that are attached to it, and they feel that they've been stretched out, and that's when you know you're full. Nobody's gonna get it.
Like synovial.
Pancreas?
Hmm?
Pancreas?
Oh, you
I
cheated.
did.
She cheated. I
Why
saw her
would you need
reading.
pressure there.
Nah, you should give her bonus points.
So, where's the pancreas? And what does it do?
Here.
Right here. I like that. It's right here.
I'm sitting down, what do you want?
It's
It has a very specific location, right? No?
next to the liver.
Your stomach sits kind of C-shaped right here, right? And then your small intestine comes out here and goes up around like that. And the pancreas sits right between them. Right between the stomach and that first section of small intestine, which is called the what again? First section of small intestine, the duodenum, right? So basically it lays right along the stomach, right? And what does it secrete? It's got an endocrine and an exocrine portion of it, right? And they both have to do with what? The endocrine portion has to do with
Insulin.
insulin and glucagon, right? Secreting those things and controlling blood sugar, right? The exocrine portion goes into the small intestine with digestive enzymes. Both of those are associated with what? Food. So when you eat, what happens to your stomach? Fills up. What's it going to do to that gland? Put pressure on it. Pressure receptors. It wasn't a big leap. It's obvious once you start thinking about it, right? All right. Pancreas, right? Where's an islet of Langerhans, by the way? Somebody put that on the last test, right? Islets of Langerhans are in the pancreas. Where do you see one? I see 1, 2, 3. 1, 2, 3. Let's see, where do I name them? 4, 5, 6, 7. A lot of them. There's tons of them in there. Where's an islet of Langerhans?
The speckly dots.
There's one. There's one. There's one. Right? Piece of cake. Easy to see. We'll come back to those. All right. Islets of Langerhans. Oh, I'm sorry. Pacinian corpuscles, right? Let's go back to the Meissner's corpuscles because now we're going to look at a Meissner's corpuscle. Meissner's corpuscles occur up here. Meissner's corpuscles occur up near this skin area up here. That's a really nice one there. Let's look at that one. So everybody can see it, I assume, right? What's this? Stratified squamous keratinized epithelium, right? You should have that down by now, right? And this is what? Irregularly arranged, dense connective tissue. Hey, look, we talked about those before, right? So a Meissner corpuscle is what's called a fine pressure receptor. Sometimes called a 2-point discrimination receptor. You don't know what that means, but we'll talk about it. When you touch something with your fingertips, right, you can feel very acutely what's going on, right? And so think about it, right? So here's one here, it's right here, by the way. I'm gonna zoom in on this in a minute, but this is a Meissner's corpuscle. So they kind of sit facing up toward the skin, right? At a 90-degree angle from it, approximately, right? So when you press on this, it presses that and causes a nerve impulse to be sent to your brain, saying, hey, I feel pressure, right? So it's a fine pressure detector, right? Called a Meissner's corpuscle. They sit in something called a dermal papillae, a skin of the dermis, or a hill of the dermis, I'm sorry. And then you have these epidermal ridges that stick down in between those dermal papillae. So what does 2-point discrimination mean? So if I took 2 needles and put them, whatever, some distance apart, and you touch them, you would be able to tell there's 2. If I move them closer and closer and closer, at some point, you won't be able to tell that there's 2, you just know that there's 1. That's 2-point discrimination. Your fingers have very good 2-point discrimination. You can be very, very close and still detect 2. Other parts of your body, they could be this far apart and you wouldn't know that there's 2, there'd just feel 1. And that's all depending on how many of these Meissner's corpuscles you have in your skin. You have them more in certain parts of your body than in others. We already talked about where you have a lot of them. You have a lot of them in your fingers, you have them in your tongue, mouth. Those places have a lot of them. Feet have a good amount of them. Where do you have the least of them? Back. Very good. Okay. Any questions on that? Let me zoom in on that and show you a better view of it real quick. It's got a very distinctive look to it. There it is there. It's very distinctive in how it looks. Everybody see it? Right here, right? Looks kind of like a stack of coins, kind of. Not really, but kind of. It's very distinctive though. It always looks like that. I'll find another one, I'll show you a different one at High Mag as well. So I'll just look around until I find another one. Or maybe I should have you stop me when you see one. That would be more fun, wouldn't it? There's one there. It's kind of a weird one. That was kind of round, doesn't it? See that one? Kind of round looking. That's probably because it's not cut clean. There's another good one in here. Let's see. Where's a good one? There's a nice one. How about that one? That's a nice one. Want to see that one? Right? Very distinctive look to them. Okay. Any questions on that? All right, let's go on to the next one. I don't know what's next. Oh, motor inflates. Okay, muscles. This is how your nerves connect to your skeletal muscles, right? This slide's a really fun one to look at. It's got 2 things that are really nice in it. So there's the nerve fiber up there. See, it looks like a tree, right? Everybody see it? The nerve fiber, see how it branches out and branches out and branches out. And they are going to end in something called the motor end plate, right? This is where you're going to have to play with focus to be able to see it, right? So if you look right here, you can see this little weird little ball shaped thing here, right? That's a motor end plate. If you look here, you can see it here. See that one? Motor end plate there. If you look over here, let's see, where's another one? There's one. That's a weird looking one there because you're looking at it on, like, down on the top of it. Here's another one here, right? So there, you have a lot of them, right? So you'll find a different spot and look at a different section of it. There's a really nice one there. I like that one. Where'd it go? Where is it? I lost it. Where'd it go? It's up there. It's up there. There it is. Okay. Here. And goes there off into that, right? So you can see the outline of it very well there. So motor end plate, say that you've seen diagrams of it in lecture already when we talked about muscle conduction and muscle contraction. The other thing you can see real well is you can see skeletal muscle really nice in here. Look at that, those cross striations in that muscle are really nice there, right? And if I zoomed in a little bit more, it'd even look better, right? I mean, those are very distinct striations in that skeletal muscle there. And plus, pretty good motor end plates, right? Anyway, that's what those are. And again, just like Meissner's corpuscles, the amount and distribution of these is dependent on where it is in your body. And that, we talked about this in lecture as well, how much fine control you have over it depends on how many of these things you have per square cubic centimeter, whatever, cubic millimeter, whatever. And I said already in lecture, what's the place in your body that has probably the highest percentage of these motor end plates, right? And I said that already. And almost all these slides, by the way, are taken from that same piece of tissue, by the way. That same organ, if you will. And where do you have the highest number of motor end plates? Tongue. Talking, right? Use it to talk all the time. Mouth as well, large numbers in the mouth, but the tongue probably has more than any other. Vocal cords are a reasonable amount of limited vocal cords, although that's, even though you use that to talk, it's just a matter of tightening or loosening it up a little bit, giving you a tone more than anything else. All right, what time is it? Any questions?
Could we maybe look at the spinal cord again?
Do I want to look at the spinal cord?
Yeah.
Why do I want to look at the spinal cord?
Like the big structures.
The big structures?
Yeah.
The golden one,
Which one?
the golden one.
We're gonna look at that next week, right?
Oh,
Yeah,
We're gonna look at all the
that.
structures in the spinal cord next week, because that's gonna be the central nervous system. Next week, we're gonna look at the brain and the two different parts of the brain, right? And we're gonna look at the spinal cord in detail with the different parts and different structures in the central spinal cord. So don't worry about anything in the central spinal cord right now. We're gonna do that next week, okay? Right now, I really only want you to concentrate on the dorsal root ganglion itself. Don't worry about the rest of it.
Okay.
Does that help a little bit? Anything else?
can we look at this section again and go over?
Yeah, that picture sucks.
Okay, fair enough.
The problem with that picture is that it's hard to focus when you get on a high mag on that cut.
Okay.
It's very hard to focus on it because you're looking at multiple different layers that all mess with each other. So what you have in there is very straightforward, though. I mean, what you're looking at, basically, right? How old are these pictures? Who drew that? I didn't draw it. That's not my artwork. How many people drew that? No? Nobody's claiming it. All right, let's see. So the picture you were talking about is the nerve mammal slide, right?
Cross section.
So what you have is you have a cross-cut axon of a nerve, right? And it usually looks kind of like that, right? So where's the axon itself?
In the middle.
That's that little thing in the middle, right? This is all myelin here. And then your endomysium is going to be right around the edges of it, right? And it will either connect to it or it will not, depending, but it should be fairly close to being connected to it, right? There might be a small gap. And then, of course, you have all the ones around it, right? And they're all over the place. Does that make more sense now?
Yeah.
Now, you will see little nuclei over here, right? Which are the nuclei of the connective tissue or the nuclei of supporting tissue that's in that bundle of some sort. So there are a few nuclei in there, but don't worry about them too much. Okay? What else you got? She's been on her phone the whole day.
I've been going back and forth because this is black and white, so I use the color version.
I printed off the color version. Anybody want my color version? You can have it if you want. I'm good. All right. Nothing else? I will not be in class tomorrow. Remember, the test is online. Get it done by Monday. Sorry I'm missing 2 days in a row. Have a good weekend. If you get stuck on something, you got lots of friends you can ask, right? Okay, have a good weekend. We'll see you.
Will you create